Hi everybody. In this video, I will show how to use Energene's visualization tool, which is based on the IGV from Broad Institute, to visualize and analyze the results of a test cross in maize. The test cross was done between real populations, and so each track here is a real population. This reel is a BSS1 times BSS2, and each line here is a single individual in this population. This part here is showing the genotype of the population, and the genotype here is showing the genotype of the first individual. A change from dark blue to light blue means a recombination event between the parental genotypes. And since the information that this genotype is based on was a very low coverage NGS data, around 0.01 coverage of the maize genome, there is uncertainty about the exact point of the recombination event, and so there's red areas. The red areas are around here are around 500 kb, and so they do not affect the actual QTL analysis. I will now switch to another tab showing the details of the cross, and we can show the analysis of the genotype versus phenotype analysis. I will now move to this tab, which shows the results of the test cross and the phenotype-genotype analysis. So in this screen here, you see a single cross population between this real population and this real population. And each two lines, so line for the first line here and the first line here, showing a single individual, which is the cross between this genome here and this genome here. And again, the color coding means that a change between yellow and dark blue means a recombination event, and here a change between light blue and orange means a recombination event. We added the phenotype to the screen. So each column here is the value of the genotype, and as you move around, you'd see the value behind the color coding. And a nice feature about the IGV is it lets you sort the whole population by the value of the phenotype. So for this example, you'd see total leaf area sorted from high to low as we go down. The nice thing about it is now when the genotype is also sorted, you can see that there are some places like here when there is correlation between the phenotype. So going down with the total leaf area means changing from orange to light blue the genotype. So this is a correlation, it's not random, and our analysis finds it. This part of the screen is showing the results of the analysis. Each line here is a phenotype, and the color coding here means the R-square correlation between the phenotype and the genotype in this genomic location. Darker the blue, the stronger the correlation. And if we move to a kind of a high-level view of the whole genome, you'd see areas where the specific phenotype is correlated to a specific genotype in a specific location area. I will now show how can you use our system to find out possible candidate genes for specific QTLs for specific phenotypes. So let's go back to the total leaf area phenotype, which is correlated to this area here in chromosome 1. And as we go here, we remember that the correlation was to these two haplotypes here, which are actually Lancaster tree based phenotype over Lancaster 1. So the actual phenotype that is considered to be responsible for the trait is Lancaster tree. Now we move back to our allele separation tab. And remember, the allele separation is showing in color coding the relations between the haplotypes of the different lines in the different areas. And we're seeing that Lancaster tree in this area has this color haplotype. And it's interesting to see that it's identical to the haplotype coming from this line, which is an iodent line. This iodent line is special because we have a very deep coverage of, And so we have a de novo assembly of the genome of this iodent, and we are able to map the B73 genome to the iodent and see in this region what are the exact sequence of the iodent. So what we'll do now is we use our capability of genome mapping, so we can switch the reference genome for a specific region and see what happens on the other reference genome. We're now looking on the B73 reference genome, and we're seeing that in this specific area, the B73 has several genes annotated, and those genes are candidate to be responsible for the QTL that we just found. Now we will move into the exact area in the iodent. Again, as I zoom in, we'll see more and more details about the genes from the B73. When we move to the iodent, we'll see first that the size of the area has changed. And now we see additional PAV genes that exist only on the iodent. This means that this PAV genes which do not appear on the B73 genome, and so would be totally ignored if we only had the previous analysis, is now a candidate to be responsible for the phenotype that we just found the QTL for, and should be considered as a candidate maybe for GM later on the trait mapping. So this is a nice example of how we combine the different parts of our system to come up with a candidate that wouldn't be found in another way. Thank you.